Hello friends, this is another tutorial video. I am explaining you as part of our online course on AI in pharmaceutical sciences. And the title for this video is De Novo Drug Designing, an original research paper which I have taken from very beautifully explained original research article written by group of scientists working in Tata Consultancy Services (TCS), world famous, genuine, ethically strong, and very close to my heart company because it is being run by Sir Ratan Tata, whose example I always give in my all sessions in all my classroom sessions. So before uh, going ahead, let me tell you I have taken the entire sentences from this paper because they have summarized very beautifully. I have taken images, infographics as such, so I'm thankful to these intelligent researchers for writing this paper and keeping this paper in public domain. Thank you very much. The title is Accelerating De Novo Drug Design Against Novel Proteins Using Deep Learning Model, and the paper has been published in Journal of Chemical Information and Modeling in 2021. I was very impressed by the way of writing and way of explaining the paper as if it reflects the philosophy of Tata's. Everything is very clear, crystal clear and transparent. So this paper ultimately moved me, and I thought to take this paper. I read this paper 3 times, understood this paper, and then I am using this paper for all the learners in this course. I am thankful to Soumya Ramaswamy Krishnan, Navneet Bang, Gopal Krishna Bulushu, and Arijit Roy sir for writing this paper and sharing their knowledge in this paper. Thank you very much. Now, without taking much of your time, I am going ahead. So on the right side, you are seeing these are various logos I have taken from the net, from their websites: Python, Campbell, Scikit-learn, NumPy, DrugBank, PDB, Jupyter, Pandas, Anaconda, Zinc, Matplotlib, and OpenBobble. So already in my another video, I have explained you. The title is various tools and technologies used in AI-driven drug discovery. There I have explained what is the role of these various tools or machine learning or Python libraries. So what are the steps in AI driven de novo drug designing? One question comes in mind: why there is need of artificial intelligence in drug designing? So let me tell you, drug discovery is one of the most stringent a different type of technology or a marathon which requires on an average 10 to 12 years, and it requires around 2.6 billion US dollar, and it is increasing year by year, but the ratio or the percentage of output is not increasing in that proportion. There is urgent need of some technology which minimizes the number of years, amount, energy, and efforts and failure. So here comes the role of deep learning models belonging to artificial intelligence category. So de novo means entirely new, novel, never before, entirely from scratch, in silico, or in the system on the screen. So the first step is, so here we are learning how to design drug like molecule through generative type of deep learning model in de novo drug designing. First step is to get the database, to get the data from database. In our case, data means chemical structures or the molecules or the compounds. So the structures are similar to drug molecule or they themselves are drug molecule. Then we will pre-process the data, that is we will curate the data, that is we will remove all unwanted, confusion causing, misleading, fake, not genuine, duplicate data, outlier, pains, or those trouble making data which is not required. It means we are removing the garbage from the data, that is we are doing pre-processing of the data, because machine learning is a technique in which if you give garbage you will get garbage. So someone famously told garbage in, garbage out. So you have to curate your data in a proper manner, right? That is called curation or pre-processing, because in in all my videos I keep telling machine learning is 80% data handling and only 20% uh, is the time devoted to the machine learning model fine tuning. So after that we will go for machine learning model selection through different machine learning algorithms, then training of machine learning models, then selection of few targets using keywords, searching of few target specific ligands. What is target? The disease causing area. For example, you are searching a molecule for lung cancer, then you have to select the targets, that is the proteins or the genes or the biological targets or receptors which are causing lung cancer. Then you will finalize few molecules which are benchmark or which are ground truth or which are available in market or which are successful blockbuster molecule successfully drugging the target. Then you will generate target specific ligand data. Then you will go for transfer learning. What is the meaning of transfer learning? I will explain in next slide. Then application of reinforcement learning for developing highly optimized drug like molecule. What is the reinforcement learning? I will tell you. Selection of promising molecule based on physicochemical features, that is you have generated few molecule through generative deep learning model, then you will choose selected molecule which are having some promising future based on physicochemical property like Lipinski's parameter and other parameters which are responsible for making a molecule drug like molecule. Removal of outliers based on certain rules, and applying of molecular modeling to endorse select molecule, that is you will go for what we call it dynamics and all those studies to check how the drug is finding a pocket in the 3D structure of the proteins. So ultimately your compound should be able to drug the receptor properly to become a successful blockbuster drug like molecule once it hit the commerce. So this is the flow chart how to go for de novo drug discovery using deep learning models. Training the generative model on Campbell or other database like Zinc database or Drug Bank or PubChem to learn the grammar of a small molecule. What is the meaning of grammar of a small molecule? That is how the drug like molecules are different from non-drug like molecule, as simple as that. Curation of data set of inhibitors. For example, you are searching a drug for lung cancer treatment, then you will search all those chemical compounds which are proven in vitro for inhibiting the receptors or gene or the protein responsible for causing lung cancer. That is called inhibitors of small molecule, inhibitors of proteins. Now you will curate the data set of inhibitors from a small molecule that modulate the activity of the same family of proteins. So for example, if you don't have the specific receptor, it is very interesting. Suppose you have a protein structure of this type. It is actual you need, but you have some protein structure which is of this type, right? So you need the ligands for this protein, but you don't have any ligand available in this world for this protein. So you will try to take those ligands which are available for a similar type of protein, this type of protein, right? So you will train your machine learning model by using the inhibitors which are fit for this protein but not for this protein. But you cannot stop the work, the show must go on. So in that case, research, it is a accepted procedure. When you don't have a receptor specific drug like molecule, then you can take the identical type of protein to decide the receptor, to decide the inhibitors. Now transfer learning with the curated data set helps focus on a particular region of the chemical space specific to the target protein of interest. Reinforcement learning to enable generation of novel small molecule with optimized docking score. Reinforcement learning is a technique in machine learning which guides the ongoing process of model training. If it gives reward to the machine learning model, it means model is going in right direction, model is learning the things which we want to teach it. If it is not learning, then we will punish it. For example, you are continuously optimizing the latent space or chemical space and you are not finding the proper binding score in a regression manner, or you are not finding proper Lipinski's parameter, then you will change your course of action. That is, you will punish the ongoing training process. It means model will learn what I am learning or what I am predicting is wrong. I have to change my way. It is called reinforcement learning, which is based on action and environment and reagent, based on reward and punishment. So after reinforcement learning, you will decide few molecules which may be promising. Then further filter down is to be done based on other physiochemical parameters and structural alerts. Rule based filter will be applied to be used to filter drug like molecule specific to the target protein of interest. In this way, you will find for example 10 molecules after transfer learning and after reinforcement learning. Then after from those 10 molecule, you will choose only 4 or 5 based on your study protocol, and you will go to preclinical or animal studies before planning for clinical trial. Okay, what is transfer learning? Suppose you don't have huge amount of data in terms of ligands. You have a structure of protein for treatment of lung cancer, but you don't have sufficient inhibitors of that particular receptor which is causing lung cancer. In that case, you will take another type of data set which is responsible for inhibiting some receptors, or a proven data set you will take from putative libraries. You will train your machine learning or deep learning model so that your model will learn what are the general common features irrespective of the disease for a particular drug molecule. So from a huge data set, you will first train your deep learning model to know the grammar or the basics of drug like molecule in general. Then this will be called pre-trained generative model. Then you will change the few hidden layer in this neural network after showing the limited type of data set which is actually required by you. This is called transfer learning. What is it? For many machine learning tasks, only small data sets are available, which might lead to overfitting with powerful models such as neural network. In this situation, transfer learning can help. Here a model is first trained on a large data set for a different task. Means data set is large, but it is not relevant to your exact project, I mean the exact protein receptor. Then the model is retrained on the smaller data set, which is also called fine tuning. Actually, you should have huge type of data for this data, but you don't have actual data, so you will train the model in a primarily manner for large data set. Then you will fine tune using a smaller data set, which is actually required by you. The aim of transfer learning is to learn general features on the bigger data set, which also might be useful for the second task in the smaller data regime. To generate focused molecule libraries, we first train on a large general set of molecules, then perform fine tuning on a smaller set of specific molecule, and after that start the sampling procedure. Sampling means you will generate different type of molecule from your generator deep learning model. For example, you have pre-trained generative model trained through this large database, and then you will go for transfer learning, and you will expose your pre-trained generative model to be trained further for target specific compounds, and you will, through this docking score, indicates how perfectly your proteins are binding or your molecule are binding to the receptor of interest. Then if docking score is not proper, you will not give reward to the machine learning model. So this cycle of punishing and rewarding goes continuously until your model learn to identify and to generate the drug like molecule. So this process is further fine tuned based on various structural alert like physico-chemical property and other parameters are there. So de novo small molecule design pipeline we will understand once again as per this paper. So first step is training the generative model on Campbell database. For example, you may choose another database also like PubChem, DrugBank or Zinc for that matter, to learn the grammar of a small molecule. Your model should learn what is required for a molecule to be called drug molecule. For this, you have to teach it what is the features in terms of descriptors, bonds, atoms, everything. Data set of inhibitors was curated from small molecules that modulate the activity of the same family of protein. Transfer learning, as I told you, transfer learning with the curated data set help focus on a particular region of the chemical space specific to the target protein of interest. Then reinforcement learning. Reinforcement learning is basically used to continuously optimize the ongoing process of generating drug-like molecule. Then physicochemical properties and structural alert were used to filter drug-like molecules specific to the target protein of interest. So in infographics, understand now, this is your source of database, Campbell. You will train a deep learning model to understand the grammar of small molecule. This model will be called pre-trained machine learning model. Now you will transfer this learning, in the sense you will expose the same model to limited set of drug molecule of interest, and then you will again go for reinforcement learning to give reward and to give penalty, and this reward and penalty optimization will force your deep learning model to generate only drug like molecules having some physicochemical properties like drugs. So this is an example of latent space. So optimize the generative model, learn from the training data set and generates molecule close to the validation data set. A low dimensional embedding of the final set of generated small molecule colored based on their docking score. The 5 small molecules 1, 2, 3, 4 and 5. The 5 small molecules generated by the model are shown according to their similarity toward either the training set or the validation set. The Campbell ID of the small molecules from the training and validation dataset is also shown. The Tanimoto coefficient of the generated small molecules with the training and validation dataset molecules is shown in the red within parentheses. See, so this is the ID of the validation set. This is the ID or the identification number of training set, and this is the Tanimoto index. So these are your molecule number 1, 2, 3, 4 and 5. So these 5 molecules have been generated by a generative model which was trained initially through deep learning model. So here in this diagram, the researchers have shown the docking score and the identification as per Campbell library. What they are telling, optimize the generative model, learn from the training data set. If they don't learn the grammar of a small molecule through training data set during training or transfer learning, then there is no use of this type of generative model. So this diagram shows the 5 molecules, how close they are to the validation data set. The 5 small molecules generated by the model, this is black structures are there, are shown according to their similarity toward either the training set blue or this is green. So this blue indicate training set and green indicate the validation set. The Campbell ID of the small molecules from the training and the validation data set is also shown. The Tanimoto coefficient, the Tanimoto coefficient of the generated small molecule with the training and validation data set is also shown in red within the parentheses. So this is the Tanimoto coefficient of 2. So you see here, it means now you have to again go to Campbell dataset and see the structure of this chemical ID. It means these structures, all these 5 structures can be tried in preclinical studies after testing their synthesis feasibility. In this way, you can use generated model to the pre-trained and trained generative model to generate various molecules which may be chemically similar to training model, or they may not be chemically similar to training model, but they have physical and chemical properties like training molecules. So in both the cases they are promising it for further studies. So this is again another infographics for same feature. So this is generative model by the team of researchers, how they developed it by training on 1.6 million drug-like molecules from Campbell database to learn the grammar of small molecule. Then transfer learning has been done for protease specific generative model, then generation of molecule based on reinforcement learning, and various criteria were selected before finalizing 3-4 molecule for preclinical testing before after synthesis feasibility. So SAS means synthetic activity score. If it is less, you cannot design that generated molecule in the laboratory, it is of no use. Then how close your molecule is to the drug like properties, and log P and molecular weight. These are the 4 essential parameters before going ahead for filtering the generated molecules, because all the generated molecule may not be drug like. So here what are the process, now we will study in detail each one by one. De novo drug discovery pipeline can be divided into the following components. As such, I have taken from this paper of TCS team. Dataset curation, pre-processing and training the generative model, as I just read from 2 infographics. Generation of target specific ligand dataset and transfer learning. Training the predictive model, then reinforcement learning for property optimized molecule generation. This is important. Already we have seen in autoencoder, property optimized means through some mechanism. For example, in this paper, scientists have used reinforcement learning type of machine learning to check in real time what are the properties from viewpoint of physicochemical features, and continuously they have to optimize those properties. Filtering the generated molecules through physicochemical properties, application of rule-based filter to remove molecules with undesirable groups, and validation of filtered molecule through molecular dynamics for confirming the binding score and everything regarding the confirmation of 3D protein and structure binding like lock and key arrangement. Now dataset curation, pre-processing and training the generative model. The first step is this. In this stage, what they have done, the first step of the pipeline is to train a generative model such that it learns the grammar of small molecules and designs novel and valid small molecule. Novel means new, not available in this world, and valid small molecule means they should be able to follow the chemistry rule and they should have good synthesis score. The data sets for training the generative model and for the case study were obtained from the Campbell database. The molecules were represented in the SMILE format to leverage the effectiveness of recurrent neural network in handling sequential data through the existing natural language processing. Already we have discussed SMILE is easy to use format for handling deep learning model, and recurrent neural network have been tested in other field also for making use of sequential data for generating more and more amount of data. The problem of learning the SMILE grammar is cast as the sequence to sequence problem, that is machine translation, and the generative model tried to emulate this problem for the SMILE dataset with stack augmented recurrent neural network. This is the basic or core structure or the architect of recurrent neural network. The SMILE's dataset was pre-processed by applying sequential filter to remove stereochemistry, salts and molecules with undesirable atoms or group. The SMILE's strings obtained were canonicalized because one SMILE can produce various more than one type of molecule. So you have to canonicalize so that only one type of structure can be made, and duplicates were removed. Only molecule with length less than or equal to 100 were collected. The RDKit library in Python, we all know, is used to pre-process the data. The generative model was trained on a dataset of close to 1.6 million SMILE string from Campbell database. The deep neural network architecture of the generative model was gated recurrent unit as the internal memory, augmented with a stack acting as the dynamic external memory. So gated recurrent unit is a specific type of feature in recurrent neural network which are famous for remembering only required information and forgetting or discarding the ones which is not required. The use of stack augmented memory enabled the generation of chemically valid smiles with high accuracy as also observed in previous studies. The trained generative model was used to generate 1 lakh compounds in 10 batches of 10,000 compounds each. This model is henceforth referred to as the pre-trained generative model by the researcher in the paper. The pre-trained generative model had a high accuracy of 96.6%, defined as the mean percentage of chemically valid molecule. So what is the yardstick? What is the measurement? What is the logic to check the success of pre-trained model? Only one parameter is there. In simple terms, easier to perform, all the generated molecule should have chemically valid molecule, and that can be checked through RDKit, which is a cheminformatic Python library. So next is generation of target specific ligand dataset and transfer learning. The drug design pipeline aims to discover novel small molecules against a specific target protein. In most cases, there is limited or no knowledge about the small molecule that can bind to target protein of interest. So you know the target protein, but you don't know what type of ligands or small molecules are fit to drug the receptor. In that case, you have to train the model using huge amount of somewhat correlated data set, but then you have to transfer the learning. In this study, an initial target specific small molecule dataset was curated, considering molecules known to inhibit a similar protein family to the target protein. In order to identify the proteins belonging to the same protein family as the target protein, the basic local alignment search tool was utilized. Then the curated dataset of small molecule from similar protein was subjected to extensive pre-processing to canonicalize and remove redundancy. It is important. You should remove all unwanted garbage data. Now, the P. Campbell score of a small molecule based on the reported IC50 values was also used to filter the dataset. Then the target selectivity of this dataset was further enhanced by docking these molecules in the active site of the protein of interest using autodock vina. I am reading because already I have discussed in earlier video, and already I have explained you in this video through various infographics, and self-explanatory language has been used by the researchers, so there is no need to go into much detail. Otherwise it would take 2 to 3 hours to explain in detail. During transfer learning, the weights of pre-trained unbiased generative model were loaded and all the layers of the deep neural network were frozen, except for the last dense layer. It means already you have a pre-trained machine learning model. You will not change the entire layers or weights of the those trained layer. Only few layers you will change or alter, or you will allow to alter based on the specific features of small data set. This allows the gradient calculation and back propagation step to alter the weights of only the final dense layer. It is important. Why you will change entire weight? Because you have purposely trained a machine learning deep learning model to learn the grammar of a small molecule. Now your model, which you have called the pre-trained model, has learned the grammar of a small molecule. You have to just change few layer just to accommodate the features of specific target specific ligands. Now you will train the predictive model. The predictive model learn a mapping between the small molecule and their corresponding experimentally determined property values, such as bioactivity or log P or others. As this work focuses on cases where no target specific dataset is available, both the high and low docking score of ligands from the previous step were used to train the predictive model. The predictive model uses gated recurrent unit as the internal memory, followed by 3 dense layer. This is the architecture of the neural network. The model was trained using a mini batch gradient descent with the Adam optimizer. Reinforcement learning, already I explained you, it involves the use of agent, environment, reward and penalty function. Property optimization, molecule design. The generative model obtained after transfer learning was combined with the predictive model to bias the generative model toward the property of interest using reinforcement learning. During training, the generative model acts as an agent and the predictive model acts as a critic. The agent learns a prior policy during training, which is the probability distribution over the different symbols at each position of the smiley string. The objective of policy gradient method is to refine or optimize this prior policy so that the reward obtained is maximum. If reward is more and more, it means your training is going toward the direction of more drug-like molecule rather to more non-drug-like molecule. So after that, you will generate various molecule and you will filter those molecules based on various physicochemical properties, and you will use few molecules for docking purposes before going ahead. So how to validate those molecules through generated through predictive model, to study the effects of solvents and also to compute binding energies. Molecular dynamics simulations were performed by this researcher using Gromex and Char 36 force field. In this way, they came to conclusion that few molecules can be further tested by synthesizing in animals and further for clinical synthesis, clinical trial, if everything goes as per the rules and regulations and in a positive manner. With this, we finish the step-by-step learning through a research paper published recently for how to train a deep learning model attached with reinforcement learning type of machine learning model for de novo drug designing for those type of disorders where you don't have the exact structure of protein and huge amount of related ligand data set. I hope you enjoyed this session. Before this, you must watch my videos on deep learning and machine learning. Thank you very much.